Hello, good day everyone. It's nice reaching out to you today on this wonderful day. You're welcome to our biology class for today. In our previous lesson, we have been looking at ways of controlling harmful microorganisms. And I made us to understand that under various ways of controlling microorganisms could actually be grouped into two. We have the physical method of controlling microorganisms. Likewise, we have the chemical methods of controlling harmful microorganisms. Under the physical method of controlling harmful microorganisms, we have looked at use of temperature, both low and high temperature. We have filtration as a physical method of controlling harmful microorganisms. We have desiccation or drying. We have the use of osmotic pressure. And in our last lab, we started looking at use of high temperature for controlling harmful microorganisms. Use of high temperature in controlling harmful microorganisms could also be called heating. And under this method of heating, we have the use of dry heat and the use of moist heat. The use of dry heat involves the use of hot air oven or the use of Bunsen burner to inoculate or maintain asepsis in the laboratory. While under the use of moist heat, we have boiling, we have the use of autoclave or autoclaving, and we have also looked at pasteurization as a method of moist heat sterilization. Today we want to continue with the topic, and today we are looking at ways of controlling harmful microorganisms still under physical method, and today we shall be looking at use of low temperature in controlling harmful microorganisms, and we shall be looking at filtration as a physical method of controlling harmful microorganisms. Now, in our previous lesson, we've been made to understand that the use of high temperature could be effective in controlling harmful microorganisms. Likewise, the use of low temperature could also be as effective as the use of high temperature, with the exception of some sacrifice which prevents cold temperature, which we were saying that the use of low temperature could be used to control several microorganisms, but it cannot be used to control sacrifice. Now, what are sacrifice? Sacrifice or psychophilic microorganisms are called loving microorganisms, particularly some species of bacteria. Their prevention temperature for growth is about 59 degrees Fahrenheit, that is about 15 degrees Celsius, which is even very low than the room temperature or ambient temperature. Psychophilic bacteria, they are actually adapted for life at cold temperature. So you cannot say you want to control the growth of psychophilic microorganism while you are refrigerating or you are freezing your substance. But for every other microorganisms, the use of low temperature could be effective in controlling the growth of some other microorganisms aside psychophilic microorganisms. Now, why? Because the essence of this psychophilic microorganism, they are unstable even at room temperature, and they fail to operate properly at room temperature, and that is why this kind of microorganism loves cold environment or cold temperature, because the cold temperature or the lower temperature is conducive for their growth or support their growth and optimum reproduction. Some sacrifice, especially those from the Antarctic, they have been found to contain some polyunsaturated fatty acid at room temperature, and this polyunsaturated fatty acid, it will become very fluid. The membrane of such bacteria will become so fluid at room temperature, which could lead to the death of this microorganism, and that's why they don't thrive well even at room temperature, but they grow very well at very low temperature. Now, refrigerators that we use in homes and in the laboratories, they have ability of maintaining temperature between 0 degrees Celsius and 7 degrees Celsius. Those are the refrigerators that we use at home and in laboratories. And this temperature of about 0 degrees to 7 degrees Celsius has ability of inhibiting microorganisms. What do we mean by inhibiting? To suppress or to reduce or to slow down the metabolic activities. And once the metabolic activities are slowed down, definitely the growth of microorganisms will be significantly reduced, which will help to preserve foods and some medical supplies. Some certain types of laboratory cultures could also be preserved by refrigerating them for later use, especially those ones that you know that you're not going to use them, keep them for a very long time. You actually want to keep for like a day or two. You could actually keep them inside the freezer at this temperature of about 0 degrees Celsius to 7 degrees Celsius, and they will keep well because the temperature will inhibit the growth of microorganisms. Now let's look at storage of bacterial culture and medical specimens that require long-term storage. Bacterial cultures and some medical specimens requiring long-term storage are often frozen at ultra-low temperature of about -70 degrees Celsius or even lower. What do we mean? You are carrying out experiment on particular species of organism or some specimen in the laboratory, and you know that you're not going to complete your experiment in a month. All you do in order to avoid contamination or other growth of microorganisms is to store this kind of specimen under ultra-low freezer. Ultra-low freezer has the ability of maintaining about -70 degrees Celsius and even lower than that. This ultra-low temperature could be achieved by storing specimen on dry ice inside ultra-low freezer. Alternatively, this specimen could be kept inside special liquid nitrogen tank. They could be kept in special liquid nitrogen tank. This special liquid nitrogen tank has the ability of maintaining temperature that is even far lower than -196 degrees C, which will have inhibited several ranges of microorganisms, thereby helping to sterilize the culture media or the specimen in question. Now, we are moving to filtration as a method or as a physical method of controlling harmful microorganisms. What is filtration? Filtration is a method of physical separating microbes from samples, and it involves the use of varying sizes of filter depending on the size of the microorganism in question. It involves is a physical separating method which involves the filtration of substance using filters of varying sizes depending on the size of the microorganism that you want to remove from the substance. Now we have various types of filters that could be used in filtration as a method of controlling harmful microorganisms. We have the use of HEPA filter, we have the use of membrane filter, we have membrane filter, we have syringe filter, we have vacuum filter. Again, types of filter includes HEPA filter, membrane filter, syringe filter, vacuum filter, etc. Now, the use of HEPA filters. The full meaning of HEPA, H-E-P-A, means high efficiency particulate air filter. H is for high, E is for efficiency, P is for particulate, and A is for air. So HEPA filter means high efficiency particulate air filter. Air is commonly filtered through the use of this HEPA filter, and this HEPA filter have effective pore size of about 0.3 microns or micrometer. Now, the symbol in bracket is used for micrometer, and it is also called micron, and this is a metric unit of measurement for length that is equal to about 0.01 millimeter or about 0.00039 inches. Very low size. The micrometer is commonly employed to measure the thickness of or diameter of microscopic organisms, just like microorganisms and some colloidal particles, because they are very small. And the size of HEPA filter is small enough to capture bacterial cells, some endospores, even some viruses. And as air passes through this filter, it will remove majority of this microorganism, bacteria, virus, and their endospores, thereby helping to sterilize or to filter or to keep the air free of microorganisms. Now, HEPA filters have a variety of applications, and they are widely used in clinical settings, in cars, airplanes, etc., even in our homes. An example of equipment that we use at home that make use of application of HEPA filter is our vacuum cleaners, the air heating and air conditioning system, the air purifier. It is the majority of this equipment mentioned. They make use of application of HEPA filter in sterilization, especially in sterilizing air. Now, another filter we're looking at is the use of membrane filter. Filtration can also be used to remove micro from liquid samples using membrane filter as a type of filter. And this membrane filter is also similar to the HEPA filter in application. The only difference is in their size. A typical membrane filter that could be used to remove bacteria have effective pores of about 0.2 micrometer, while I told us that the HEPA filter has about the pore size of about 0.3 micrometer. Now this one has a filter size, pore size of about 0.2 micrometer, and this size is even smaller than the average size of a bacteria, because the average size of a bacteria is about 1 micrometer. So which means any sample that is passed through this membrane filter definitely will prevent the passage of bacteria with the sample. Membrane filtration is used for removing bacteria from various heat-sensitive solutions that are used in the laboratory, like the antibiotic solution and the vitamin solution. Like the antibiotics, if you want to sterilize them and you use autoclave, the heat that will be produced by the autoclave is going to denature some substance in this antibiotic. Don't forget, I've told us that antibiotics are also certain types of microorganisms, but that have ability of inhibiting the growth of other microorganisms. Now, if you're sterilizing this kind of antibiotics, the heat is going to denature the protein, thereby and tampering with the quality of the antibiotics, which will not give you the effectiveness of the antibiotics or some vitamin solution. And that is why they are not usually sterilized using heat, but what you do is to filter them. They're usually sterilized through the process of filtration by passing them through the membrane filter in order to remove some bacterial content in the samples. Now, large volume of culture media may also be sterilized rather than autoclaving in order to protect heat-sensitive components. And when you are filtering smaller content, instead of using membrane filter, what you use is the syringe filter. The syringe filter is just like the normal syringe, but I have a filter at the mouth, whereby the content will pass through the filter before coming out of the needle. That is the syringe filter, which is usually used for sterilizing smaller substance. But when you are filtering or sterilizing larger quantity through the use of filtration, you make use of the vacuum filters. That is where we're going to end the class for today. Don't forget, in our class today, we have looked at the use of low temperature in sterilizing or controlling harmful microorganisms. And I've told us that the refrigerators that we use in the homes or in our laboratory have ability of maintaining temperature of about 0 degrees Celsius to 7 degrees Celsius. From 0 degrees C to 7 degrees, and this temperature will have inhibited the growth or slow down the growth of microorganisms. While also I told us that certain bacterial culture or medical specimen that require long-term storage could be frozen under ultra low temperature of about -70 degrees Celsius or lower than that, and this could be achieved by storing specimen on dry ice in ultra low freezer, alternatively in a special liquid nitrogen tank. And this special liquid nitrogen tank has ability of maintaining temperature of about -196 degrees Celsius, thereby helping to sterilize or helping to control the growth of various range of microorganisms. We have also looked at filtration as a physical method of controlling harmful microorganisms, and I've mentioned types of filter as HEPA filter, membrane filter, syringe filter, and vacuum filter. And I've told us the full meaning of HEPA, that is high efficiency particulate air filter, with the pore size of about 0.3 micrometer. While we have looked at the use of membrane filter. Membrane filter also is similar to the use of HEPA filter, but it has effective pore size of about 0.2 micrometer, which is even smaller than the size of a bacteria. And I told us that the average size of a bacterium is 1 micrometer. And I told us that you could use syringe filter in order to sterilize smaller quantity, but when you are sterilizing larger quantity of substance or samples, you make use of vacuum filter. And that is where we end the class for today. See you any other time. Have a wonderful day.